Hello, everyone. We will wait for 2 minutes for attendees to enter the Zoom meeting room. And we will start about 2 minutes later. Hi, everyone. Welcome to this webinar tonight. I'm Mingkun Zhang, a scientist from R&D department of the GemPharmatech company. And I'm glad to have the opportunity to share with you some information about the FcγR receptors and their roles in the immune therapies. I believe that most of us saw this news last month. Affimed presented their updated clinical data from phase 1 or 2 study of AFM13 at the AACR 2022 conference. The combination use of AFM13 and cord blood-derived NK cell resulted in 100% response rate and 62% rate of complete responses, which is the highest response rate reported so far in Hodgkin lymphoma patients with a treatment refractory disease. The response data is quite impressive. As you can tell from this figure, AFM13 is a tetravalent bispecific antibody designed for treatment of CD30 positive malignancies. AFM13 works as a bridge that connects the NK cell and tumor cells, binding to CD30 on the tumor cell surface with 2 arms, and the CD16A on NK cell surface with the other arms. The binding will bring the NK cell and tumor cells together and form an immunological synapse. NK cell will be activated by the antibody binding and release perforins and granzyme to destroy the target lymphoma cells. CD16A used in this drug is an important activation FcγR receptor on NK cells and also expressed on macrophages and monocytes, which are both powerful weapons of the innate immune system. FcγR receptors are the binding targets for Fc region of IgG. However, as indicated by the design of AFM13. They can also be selected as targets for the FAB binding to activate the innate immune cells and precisely kill the tumor cells. Today, we're going to talk more about Fc-gamma receptors and their roles in immunotherapies. This is a diagram for a well-established tumor. In most cases, the tumor microenvironment is immunosuppressive. Multiple mechanisms are involved in generating and maintaining of the immunosuppressed tumor microenvironment, which will facilitate immune escape, thereby driving tumor progression and metastasis formation. As we can see here, many different immune cells are infiltrated in the tumor microenvironment, including adaptive immune cells like T cell, B cell, and B cell, and some CD8 and Treg cells, as well as innate immune cells like MDSC, granulocytes, and NK cells. Some of the cells are suppressive cells, such as Treg, MDSC in the left figure, and also indicated as blue color in this figure. While the others are effector cells like CTL, NK cells, and also DC and macrophages. But most of the effector cells in the microenvironment are silent and do not attack the tumor cells. In total, they together make up the tumor complex. The immune cells in the tumor microenvironment are not standard alone. They interact with each other and even interact with tumor cells through different signal pathways. The majority of these pathways are mediated by receptors and ligand binding, like cytokines and receptors, such as IL-15 binding to IL-15 receptor, and also checkpoint molecule, just like 4-1BB ligand binding to 4-1BB in the NK cells and T cells. And also MHC binding with TCR. Among of those pathways, Fc and Fc-gamma receptors engagement is one of the most efficient and complicated pathways that are involved in the antibody-dependent cell responses, like ADCC, ADCP, immune cell activation, and cross-linking. Taking NK cell as an example, the activation Fc-gamma receptor 3A engaged by Fc region of IgG will trigger NK cell activation and killing. Fc-gamma receptor mediated cell response is one of the working mechanisms of many therapeutic antibodies. And Fc-gamma receptor are expressed by many different immune cells. This diagram shows the corresponding events associated with the binding of IgG to different Fc-gamma receptors. For example, the binding of antibody with soluble antigen results in the formation of immune complexes, which will be further engaged by Fc-gamma receptors on the immune cells, leading to the events such as enhanced innate immune cell activation, target cell killing, and also enhanced phagocytosis and enhance the presentation of antigens to T cells. Specifically, engagement of activation receptors CD16A on NK cell surface with IgG results in antibody-dependent cell-mediated killing of antibody-coded targeted cells like this one. Well, by contrast, FcγR receptor 2B is the only inhibitory FcγR receptor subclass. The cross-linking of this receptor with other targeting binding antibodies will enhance the co-stimulation signals of some TNFR superfamily members like CD40, 41BB, and also OX40. So CD32B is often applied for cross-linking in many therapeutic antibody designs. Well, CD16A is the most well-known receptor for its ability to direct ADCC and ADCP. And during the drug development process, the efficacy and safety of the therapeutic antibodies will be carefully evaluated in the preclinical stage. Here, taking the CD20 targeting antibody as an example, CD20 is a promising target for many ADCC-based therapies to treat B-cell lymphoma, like rituximab. This antibody binds to the CD20 with FAB, while engage with CD16A on NK cell surface, resulting NK cell activation and eventually kill the CD20 positive tumor cells. However, rituximab was proven to be less efficient in patients with low affinity FcγR receptor 3A. There are different SMP type of FcγR receptor 3A. Because of this issue, another CD20 antibody, obinutuzumab, was developed to address this issue. Rituximab and obinutuzumab are both commercially available CD20 targeting antibodies working through ADCC. Rituximab contains a wild-type human IgG heavy chain constant region, while obinutuzumab contains a glyco-engineered IgG1 heavy chain constant region. The engineering of the Fc region will lead to enhance of the binding capacity to FcγR receptor 3A, which will result in enhanced ADCC activity. Clinical data in patients had demonstrated that the Fc-enhanced obinutuzumab confers a higher response rate and a survival advantage over rituximab. Well, did the different performance predicted in the preclinical studies? Here is as indicated in this slide. This is a head-to-head comparison preclinical study of rituximab and obinutuzumab. In this study, two types of animals are used. The first animal used is in this study is RAG2 knockout mice carrying the wild-type FcγR receptor, wild-type mouse FcγR receptor. RAG2 gene was knocked out to allow xenograftment of human tumors to this animal. And the second type of animals are RHUFR. The generation of this animal is demonstrated in this figure, which is produced by crossing the RAG2 KO mouse with a transgenic mouse carrying human FcγR receptors. And luciferase targeted tumor cells were inoculated in these mice, as you can see from figure B at the top right corner. When compared to the untreated and isotype group, rituximab and obinutuzumab inhibited the tumor growth at a comparable level in the FcγR receptor wild type group. However, the same treatment inhibited tumor growth more significantly in FcγR receptor humanized mouse group. And the most important observation is that obinutuzumab confers higher tumor inhibition than rituximab in the FcγR receptor humanized mouse model, but not in the wild type mouse. You can see the inhibition rate is equivalent between these two groups. And you can see here, all the animals were tumor-free in the obinutuzumab treated group, and a more detailed description you can see is from figure C. Both the tumor volume and the survival rate, survival curve are much better when treated with obinutuzumab, which is consistent with the clinical performance of these antibodies. This data indicated that the differences of responses from Fc-engineered antibodies can only be observed when the human FcγR receptors are existed in the preclinical animals. When evaluate human antibodies in the animals, then why there is a difference? The first factor is here. Actually, the FcγR receptors are different between mice and humans. As you can see in this figure, there are 7 FcγR receptors in human. Actually, there are FcγR receptor 1A and 1B, and also many subtypes for the other receptors. However, there are only 4 in mice. Further, we cannot find some human orthologs in mice, such as FcγR receptor 2A, 2C, and also 3B are only existed in humans, but not in mice. And also FcγR receptor 4 existed only in mice, but not in humans. Furthermore, the expression pattern of the human and mouse genes are quite different. As you can see from this table, there are 2 FcγR receptor 3 genes in human, 3A and 3B. And 3A is expressed only on NK cells and macrophages, but not on neutrophils. You can see here, no expression on the neutrophils. But FcγR receptor 3B is expressed restricted on neutrophils and basophils. However, there is only 1 FcγR receptor 3 gene in mice, which will express across different immune cell population. Expression difference also exists on other members such as FcγR receptor 1 and also 2B. The FcγR receptor 4 in mice is considered to be the equivalent ortholog for human FcγR receptor 3A, but it does not express on NK cells. such as the expression of FcγR receptor 3A in human. Last but not least, the binding affinity of human IgG to mouse Fc-gamma receptors are different from mouse IgG. As we can tell from this figure, although human IgG1 can bind to a mouse Fc-gamma receptor 4 and mouse Fc-gamma receptor 1, the affinity is lower than the mouse IgG2A binding to these receptors. And also you can see mouse IgG1 does not bind to Fc-gamma receptor 1. There are many differences. And taking together, the different responses of human therapeutic antibodies in the preclinical animal studies might be attributed to the difference in Fc-gamma receptors from the genes, the affinity, and the expression pattern. And also, human Fc-gamma receptors are required for a real response. And based on all of this background information, we think it's apparently necessary to put human Fc-gamma receptors into mice. Then, how to create an ideal animal model for the Fc-gamma receptors? Regarding the discrepancy of Fc-gamma receptors between mice and humans, we decided to put all the human Fc-gamma receptor members into mouse genome to establish the Fc-gamma receptor fully humanized mouse model. The mouse endogenous Fc-gamma receptor genes were knocked out. In order to retain the human genes as well as the human native regulatory elements, we fused several human genomic fragments including all the human Fc-gamma receptor members into one large bacterial artificial chromosome, which called BAC for abbreviation. And the large construct will injected to the zygotes derived from B6 background mice. The founder mice will selected by PCR and molecular validation. Animals stably carrying the entire bag are selected as founders to cross with the mouse Fc-gamma receptor 1 and Fc-gamma receptor 2, 3, and 4 knockout strain to establish the final Fc-gamma receptor fully humanized mouse model. So here is the brief strategy to show the genetic background and composition of the Fc-gamma receptor transgenic mouse generated by our company. Because all of the Fc-gamma receptors are expressed on immune cell surface, we analyzed the expression of human and mouse Fc-gamma receptors by flow cytometry. And as expected, the expression pattern is consistent with that of human Fc-gamma receptors expression in human, rather than the expression pattern of mouse. For example, mouse CD32 does not express on neutrophils, but the human CD32 was reported to express on neutrophils. And the results here in our mice indicates the expression pattern of the transgenic mice follows the expression pattern of its expression in human. Also, the expression of human CD16A, which is the important receptor selected for ADCC, can only be detected on the surface of NK cells, but not neutrophils, which is also consistent with the expression pattern of CD16A in humans. Since we do not have CD16B specific antibody, we also used an antibody that did not discriminate Fc-gamma receptor 3A, CD16A, and CD16B for flow assay. And as you can see, CD16 expression could be detected on both neutrophils and NK cells, which means human Fc-gamma receptor 3A and 3B, which is also called CD16A and CD16B, were expressed following a human pattern. Because there are very few macrophages in the blood and spleen, we collected the peritoneal exudate macrophages, which is called PEM, from the mouse, the wild type, and the Fc-gamma receptor transgenic. This is heterozygotes, which still carry the wild type Fc-gamma receptors. We use the PEMs for CD16A expression assay. And as you can tell from this figure, a profoundly expression of human CD16A can be detected on the cell surface. In a preliminary in vitro ADCC experiment using NK cells derived from the Fc-gamma receptor humanized mice, we culture the NK cells from the transgenic mice and also from wild-type B6 mice with MC38 cells, which was engineered to express human PD-L1 on the cell surface, and treated with avelumab, which is anti-PD-L1 therapeutic drug. As you can see from here, abveris target tumor lysis can be achieved by PD-1 antibody-directed ADCC as compared to the wild-type B6 control. We also carried out an anti-tumor efficacy study in the Fc-gamma receptor fully humanized mouse model. Drug A and drug B are therapeutic antibodies against the same target, but in a different structure. Both drug A and drug B work depending on ADCC. And the MC38 tumor cell line was engineered to express human antigen on the cell surface. The engineered tumor cells are transplanted to the Fc-gamma receptor humanized mice, and the mice were treated with IgG isotype or indicated antibodies. And as demonstrated in this figure, both antibodies are able to inhibit tumor growth, and drug B has a relatively stronger capacity to kill the tumor cells than drug A. Overall, these data indicate that the Fc-gamma receptor fully humanized mouse model can express human Fc-gamma receptors following the human pattern. And the human receptors expressed in the humanized mouse model can work as expected in preclinical studies. On top of the efficacy study, the adverse event of therapeutic antibodies should be considered seriously as well. As we know from the clinical response, many different immune-related adverse effects observed in antibody treatment, specifically the immune checkpoint related antibodies, such as liver damage and also damage to other organs like lungs, hearts, kidneys, and so on. The 4-1BB agonistic antibody, urelumab, has been proven to strongly inhibit tumor growth in clinical use. However, as we can see from this table, in accompanying the therapeutic benefits, a higher incidence of treatment-related adverse events are also observed in the clinical practice. In particular, as you can tell from this table, liver toxicity is highly correlated with treatment doses. You can see there are highly incidence in the dose of over 1 mpk. 4-1BB is a co-stimulator on T cells, which has attracted many interests in drug development in our days. Some of the strong agonists, like the 3H3 used in this study, were strongly inhibited tumor growth in the animal study. Well, weak agonists like LOB 12.3 used in this study were shown to have very limited toxicity. As you can see in different tumor-bearing animal models, both of the drugs can inhibit the tumor growth significantly, while the strong agonists 3H3 will also leads to liver damage in both the tumor-bearing animals and naive animals without tumor. Also, in the histology figure, as you can see here, pointed by the yellow arrow, highly immune cell infiltration can be observed in the 3H3 treatment group. To figure out the key factors involved in the efficacy and toxicity, an in vitro assays was carried out as demonstrated in this figure. Purified CD8 T cells were stimulated with anti-CD3 and indicated 4-1BB antibodies. And co-cultured with certain mouse FcγR receptor expression cells. These cells are engineered to express one kind of FcγR receptor on the surface. And because the cells are not immune cells, so engagement of the Fc with the FcγR receptor will not activate the cells. And as you can see from these figures, when the FcγR receptors like FcγR receptor 3 and 2b was present, the co-stimulatory ability of 4-1BB antibodies was enhanced, indicated by the increased release level of interferon gamma as compared to the control group. which means the Fc gamma receptor cross-linking will enhance the activity of antibody against anti-4-1BB. Well, the results were consistent when coming to the in vivo circumstance. When the Fc gamma receptor 2B was absent in mice, the tumor inhibition ability was reduced for both 3H3 and LOB 12.3, and you can see almost abrogated in the LOB 12.3. However, deficient in Fc gamma receptor 3 or other receptors will not lead to the similar reduction. Further, when the mouse IgG2A, which selectively bind to the activation Fc gamma receptors, was used instead of the Fc gamma receptor 1, the tumor inhibition ability was also reduced. And these results indicate that cross-linking by Fc gamma receptor 2B will enhance the co-stimulation ability of the 4-1BB antibodies. However, binding to the activation Fc gamma receptors by IgG2A isotype will compromise the antibody's activity. Consistent with the compromise of activity by activation Fc gamma receptors engagement, preventing the binding of strong agonistic antibody such as 3H3 in this study to Fc gamma receptors, to the activation Fc gamma receptors like Fc gamma receptor 3 and also other activation receptors will increase the ALT level, which is a liver damage indicator, the results indicate the higher liver toxicity. As you can tell from this figure, when Fc gamma receptor 3 or other activation Fc gamma receptors are absent, the toxicity of a strong agonist will increase. And the similar results can be observed when using the DANA mutation, which is Fc silent mutation antibodies. But the increase of ALT level by the treatment of weak agonistic antibody will not observed in these animals. So actually, the liver toxicity observed in the Fc gamma receptor deficient and intact animals indicate the toxicity was determined by both the FAB and the FC region of the 4-1BB antibodies. Based on the observation, here's an optimizing strategy for 4-1BB therapeutic antibody development. A strong agonistic antibody like this one can co-stimulate T cells when Fc gamma receptors are absent, while a weak anti-4-1BB antibody require Fc gamma receptor mediated cross-linking for its activity. When 4-1BB antibodies are cross-linked by activation Fc gamma receptors, ADCC or ADCP will compromise the co-stimulation ability. When 4-1BB antibodies are cross-linked by FcγR receptor 2B, it will not cross the ADCC or ADCP and induce strong T cell activation. Therefore, when strong agonists are engineered with higher binding affinity to activation FcγR receptor and lower binding affinity to the inhibitory receptors, it can cause superactivation of T cells and therefore induce liver toxicity. While the safe therapeutic antibody in this case is relatively narrow. When weak agonistic antibodies are equipped with FcγR receptor 2B specific binding Fc region, there would be a comparably wide window for potent anti-tumor activity and desired lower liver safety, liver toxicity profile. Theoretically, the efficacy and the safety of human therapeutic antibodies should be evaluated directly in preclinical animal studies. The target's humanized mouse model, like a CD137 humanized mice, were already created for efficacy study purpose. And as you can see from this figure, a significant tumor inhibition could be observed in response to the treatment with 40 human urelumab. Also, if the Fc part of the fully human antibody was swapped to the mouse IgG1, the anti-tumor effect will be further enhanced. While if the Fc was mutated, the therapeutic effect of the antibody will reduce. On top of the significant tumor inhibition activity, the severe liver toxicity observed in the clinical trials of the fully human antibody urelumab was not recapitulated in the CD137 humanized mice. And as you can see here, if the fully human antibody was used to treat the animals, just a mild increase of ALT level was observed. Considering the toxicity was attributed to the FcγR receptor engagement, the Fc of urelumab was replaced with its mouse counterpart, mouse IgG1. As you can see here, once the mouse IgG1 format of urelumab was used to treat the mice, significant elevation of ALT level was observed when treating with using this antibody. Similar results was observed when using the fully mouse format of antibody. Also, the histology results indicate a higher immune cell infiltration in the 3H3 treated group, which is the strong agonistic antibody. overall, as we can see from this information, we can get a brief conclusion. If we want to predict the real response of human antibody in the preclinical studies, besides the human targets binding by Fab of antibodies, there should be the 40 human Fc-gamma receptors present in the same mouse model to make sure the Fc-gamma receptor and Fc region of the antibody can perfectly matched to direct real responses from multiple immune cells. And besides the Fc-gamma receptor humanized animals, we also have many other models. And for the latest update of the status of this animal, we have some live animals in-house ready for sale. And the cohort will reach over 100 females from July this year. We also accepting pre-orders, and also collaborative characterization opportunity is opening. We also developed many other co-stimulation targets on top of the Fc-gamma receptor humanized background, which will provide profoundly research tools for preclinical studies of these targets. Okay, thank you all for attending. If you have any questions, I'll be happy to have a discussion with you. Well, there is a question. Is there a genetic evidence linking Fc-gamma receptor biology to immune cell activation? We carried out in vitro ADCC study, and also the in vivo study will provide some evidence for the activation, but we still need to collect some directly activation evidence for different Fc region, for different isotype against the different Fc-gamma receptors on immune cells. And the data collection is still in progress. If you have any suggestion, we will be happy to know, and we'll get more validation data to update in the stream information and present this data with to you. Well, there's another question. What about having a human neonatal Fc receptor there as well? Well, I think that will much better. Will be much better because FcRn is also important component to recapitulate the human antibody half-life, the recycling of human antibody in the preclinical animals. We also have FcgR in humanized mice, but still not incorporated with the Fc gamma receptors fully humanized mouse model. But that's a good point. Thank you very much. Maybe we will incorporate these gene humanized mouse model together to get a really fully Fc receptor humanized model. Well, if there is no more question, thank you again for attending this webinar.